Good afternoon, my name is Dr. Chris Arthur. I'm here at the School of Chemistry at the University of Bristol. This is the Mass Spectrometry facility where we help scientists from across the university and outside get information on their samples. In Mass Spectrometry, we are measuring the molecular weight of molecules, and from that information we're going to work out identity of the molecule, and it's going to help us understand what's inside our sample. At post 16, you're interested in time of flight mass spectrometers. Okay? This is a time of flight mass spectrometer in front of me. So the way that we analyse our molecules in a time of flight mass spectrometer is first we've got to generate an ion of our molecules. Now, we do that on this instrument by a process called molding, where we shine a laser onto our samples. That gives us a plume of ions that we can ionize and then analyze. Molding is a solid phase technique. Okay? So we have a target plate here made of stainless steel, and on the target plate we have a number of sample positions where we can deposit a sample of our compound of interest along with a molecule that we call the matrix. So the matrix is an aromatic acid. We'll then leave this solution to dry, and we get crystals of our analyte and of our matrix all mixed up together, and then we use the laser inside the instrument to illuminate the sample and generate a plume of our ions. We've opened up the instrument so that we can see what's going on inside. Now, the first thing that we need to know about a mass spectrometer is that there's a vacuum inside the instrument. The vacuum is there to stop the analyte ions that we're interested in measuring the mass of from hitting air and being knocked about. So the first thing we've got to do is load our sample into the instrument, but without breaking the vacuum that's inside the instrument. So the first thing we've got is this airlock at the front, and all I'm going to do is I'm going to put the sample plate inside this airlock. So I take this, I open it all the way up, all the way down, and I can slide the sample plate into the instrument just like that. Okay. That will now load the plate into the path of our laser. The laser can be seen at the rear of this instrument here, in this blue box. The laser beam itself will pass through a series of mirrors and lenses onto our sample plate, where it will heat up the spot, causing our matrix and our analyte to turn into a gas. Our matrix, because it's an acid, will protonate our analyte molecule, and that will give us the ion, the molecule that we're going to analyze. This plume of gas, analyte gas, will then travel down here until we reach this box here. Inside here, there's a very short electric field. Now, as you may be aware from your studies, a charged particle moving inside an electric field will experience a force. And we're going to take advantage of that. By passing through this small electric field, we give all the ions the same kinetic energy, the same energy of motion. Now, kinetic energy is given by the equation 1/2 times mass times velocity squared. Our kinetic energy is fixed by passing it through this electric potential. Our mass is fixed, that's what we're measuring, so our velocity has to change. Now, velocity, being distance over time, means that if we give it a fixed distance, i.e. this time of flight chamber here, then all we have to do is measure the time it takes for ions to travel up here. Now, for a relatively small ion, that speed could be of the order of about 30 kilometers per second. So we can time this ion incredibly precisely as it travels up here. Now, heavier ions will be slower than small ions. So the small ions will be much, much faster, and we just time them. And that's the basis of time of flight mass spectrometry. We've loaded our sample into the mass spectrometer. Okay, and we've got the control computer here, which will give you a quick guided tour of. So at the top, we've got a little camera that's going to show us our sample on the plate, just to make sure that it looks okay. We've got a little map of the plate that's inside the instrument, which allows us to select the sample that we're going to run. I'm just going to come across and I'm just going to click on the position where I put some sample on today. And you can see your sample underneath that in the little camera there. And here we've got where our spectrum's going to appear. It's got two axes. Along the bottom we've got our mass to charge ratio, and along the top here we've got our intensity axis. So what we'll see in a moment is we'll see the peaks that are detected, the ions that are being detected, appear on our mass spectrum. And it's a really straightforward instrument for us to use. We have control over our laser power that we're going to use. We have control over things like our mass range, and then ultimately we've got a big start button to start us off. So I'm going to come across here and I'm going to press the start button. What you'll see is the laser flashing on our sample as it records the data. So I'm just pressing the start button now. You see the laser flashing on the surface of our start button, and we see our mass spectrum of our sample appearing in our spectral window on the right. You see that we have principally one compound in here. We have our molecular weight that appears on that as we measured it. So this is currently 904.45 atomic mass units. I can click and zoom in on this, and if we zoom in close enough, then you'll see that that peak isn't actually just one peak, but it's a cluster of peaks that are all separated by one atomic mass unit. So I have 904, 905, 906, and 907. So what do you think those peaks might be? So if you said isotopes, then you're absolutely right. The molecules that you're used to looking at, perhaps caffeine or something like that, you'll have a very small isotope peak for the first of the isotopes. As you become bigger and bigger, or as your molecules become bigger and bigger, then the role of isotopes in your mass spectrum becomes even more important. And what the mass spectrometer is able to do is to separate those isotopes from one another. There are two reasons. So every element and every isotope has a unique mass associated with it. And what that means for us is if we can measure the molecular weight of a molecule precisely enough, then we can work out the elemental composition of that molecule. And from that information, we can help determine what it is. Now, there's lots of other advantages to mass spectrometry compared to other analytical techniques that you have encountered. NMR, nuclear magnetic resonance, works on largely pure compounds, and you need quite a lot of that compound in order to do the analysis. This instrument is incredibly sensitive. So we are putting a fraction of a microliter of material on a plate of a concentration that might be only nanomolar or picomolar concentrations of that amount. And then with our laser, we're getting even fewer ions. So we're probably only talking about of the order of maybe 100 million ions that we're detecting. Okay, so it's an incredible amount of sensitivity. Mass spectrometers can also be coupled to chromatographic instruments. That allows us to not only look at molecules that are incredibly low in abundance, but it allows us to look at complicated mixtures. That's really important if, for instance, you're wanting to analyse bloods, or you're wanting to analyse an environmental sample, say it's seawater, or it's soil analysis that you're performing, or you're looking at, say, drug doping. This instrument, because it's using a laser to study its samples, we can measure spectra not only with mass spectral resolution, but also with spatial resolution. And that's an idea called multi-imaging. Okay, so we can take a mass spectral image of a object. Okay, so we've had, for instance, sections of tissue in here, so that you can say where's the healthy tissue, where's the diseased or sick tissue. Instruments like this have been developed for use in surgery, so that a surgeon could, for instance, be cutting away a tumour, for instance, and have an idea of we have healthy material, we have sick material, so that the surgeon doesn't have to remove more than they need to. The smallest mass that this will measure is 5 daltons, so 5 atomic mass units. The largest thing that this instrument will analyse is 500,000 atomic mass units. Okay? So that's the size of a large protein. So this instrument's about 5 years old. When it was bought, it was about £600,000. Different universities will allow different access to instrumentation. So here at Bristol, because of the importance of mass spectrometry, there are a number of mass spectrometers in the teaching labs that undergraduates have access. There are also a number of experiments in their third year here where they come into this lab and they'll perform experiments under supervision on these instruments, so they can get hand-on experience with state-of-the-art equipment. Mass spectrometers are really robust generally. Okay? The oldest instrument we've had in the lab was just decommissioned recently, and that had a lifespan of about 25 years. This is one of the noisiest labs in the whole of the university. And the reason for that is all mass spectrometers have got to be operated under a vacuum. Okay, so we do that so that when we generated our ions, the ions, and they are moving through the instrument, they don't hit into molecules inside air, so nitrogen or oxygen gas, typically, and then become deflected or affected by that. So all mass spectrometers operate under a vacuum. And the noise that you can hear in the background is the vacuum pumps that we've got running all the time in order to suck the air out of our instrumentation. It's also colder in here because the room is air conditioned. And we do that because when we've been talking about time of flight mass spectrometers, we know that velocity is distance over time. And we're measuring the distance, which is our time of flight's chamber length. If that distance changes through thermal expansion or contraction, then our time measurements and our velocity measurements are going to be wrong. The instruments here are carefully calibrated for a room temperature that we've specified. Okay? Hence why the jumper. Lots of different mass spectrometers use different gases for a number of different processes. So the most common gas we use in here is nitrogen, which we use for electrospray ionization to create that very fine mist of droplets, which we generate our ions from. This instrument will also use argon or helium so that we can deliberately break molecules in the mass spectrometer. The process is called fragmentation. Okay? And when we fragment a molecule, molecules fragment along the weakest bonds within the molecule. And from that information, we can get structural information on the molecule that can help us identify what's inside. We could use that in a number of different ways. We could, for instance, search a library of known compounds. So you may be familiar with this if you've watched crime programs typically, and they'll identify a molecule based on its fragmentation pattern. That kind of information helps us identify known compounds that are within the samples. We could also use it in combination with other techniques to help us work out the structure of unknown molecules from that fragmentation information. We don't use air because of the oxygen that's in the air. Okay? When we generate an ion, that ion has a lot of energy associated with it. And if we hit that into an oxygen molecule, then we can start to see that molecule react with the oxygen that's inside there. So the mass of a molecule that we ultimately measure won't be the same. The lifespan of a detector is typically about 5 years or so. There are other parts of the instrument that accumulate material a lot faster, particularly things like the lenses that focus the ions into the instrument. I've been involved in a wide range of different projects that involve mass spectrometry. Basically anything where the scientist or researcher wants to know what's in that sample. Okay? So we've used mass spectrometry to, for instance, study healthy and sick tissues. So we can take tissue sections on this MALDI instrument of, for instance, brain sections with brain tumours. By using the mass spectrometer, we can see the healthy and the sick tissue side by side on the slide. So that helps us and helps the scientists involved to understand the molecular basis of the disease and the progression of things like tumour growth. Okay? I've also been involved in various archaeological projects over the years. So one of my favourite ones of recent years was studying some bits of bone that had been collected during the recent excavation work at Bristol Royal Infirmary. These bits of bone had come from a mass grave from a number of centuries ago, and we were asked what had these people died of. So we were able to look at the remains of the proteins that were within those samples and to effectively diagnose those patients with tuberculosis hundreds of years after they died. This kind of experiment really speaks to me because it's using mass spectrometry to illuminate part of our shared history. Okay? And I love that idea. I'm also involved in a project at the minute looking at how carbon is sequestered by our oceans. So what are the chemical processes that are going on that are trapping carbon dioxide from our atmosphere and helping tackle climate change? So really using mass spectrometry to understand those geochemical processes so that we've got a better understanding of where we might be going with climate change. The smallest mass that this will measure is 5 daltons, so 5 atomic mass units. Okay, so that will allow you to look at most atoms. And if I was to turn the laser power up onto the stainless steel plate, you'll see peaks corresponding to the metals that are inside your stainless steel plate. Okay? So that's the smallest. The largest thing that this instrument will analyze is 500,000 atomic mass units. Okay? So that's the size of a large protein or protein complex. So, for instance, iron is moved around your body by a protein called transferritin, and transferritin forms large complexes of itself, and those are about the size that we've got here. It is possible with time-of-flight mass spectrometers to go even bigger than that. In principle, there's nothing limiting the mass that these have. So some of the state-of-the-art experiments in mass spectrometry have measured the weights of, for instance, entire viruses. That becomes important because, for instance, molecular changes, for instance, in a virus that you might be able to detect by mass spectrometry might be indicative of, for instance, a new strain that's emerging. So this instrument's about 5 years old. When it was bought, it was about £600,000. Mass spectrometers at the higher end of what mass spectrometers do can be very, very expensive. So if you're looking at, for instance, carbon dating, which you do by mass spectrometry, okay, those mass spectrometers are the order of about £1.5 million in order to buy. The other end of that are small benchtop or desktop mass spectrometers, which don't give you the mass spectral resolution that you might like, but are very good in various applications. So you might have encountered these before. So when you go to the airport and they, for instance, swab down your bags as you're going through security, and then they take it away and they put it into a machine, that's a little mass spectrometer there that's really just looking for a few specific compounds, types of explosives and various drugs. Okay? So from that little swab, they're able to see if you might be carrying that. Those are much cheaper and much more sort of commercially priced. In mass spectrometry, we take advantage of charge on a molecule in order to both detect the molecule and to manipulate that ion. In this case, in time of flight, we're accelerating the ion. But in other types of mass spectrometry, we might be, for instance, curving the path of the ion. The fact that it's charged means that we're not measuring mass, we're measuring mass to charge ratio. Now, most of the time, the charge will typically be 1. Okay? However, the bigger a molecule gets, the more sites it has that can possibly become charged. Okay? So, multiple charging, so values of charge that are greater than 1, are possible. Okay? And we take advantage of that, particularly when we are doing the mass spectrometry of proteins or polymers, where the multiple charging allows us to essentially analyze the molecules that are too big for the mass spectrometer. Mass spectrometers have got to be able to do 3 jobs. The first of these is to generate ions. The second is to separate those ions by their mass to charge ratio. And the third is to detect those ions. There are a number of different ways by which we can generate ions in mass spectrometry. In MALDI, we're using a laser. The other most common way of generating ions that you might encounter is electrospray ionization. In electrospray ionization, we spray a fine mist of droplets of our sample. And these droplets, as the solvent that's in the droplets evaporates, we're left with our charged molecule. Now that's incredibly powerful if we're wanting to analyse very large molecules, where we can essentially increase the amount of charge we have on the molecule. Now we might be talking about ionization of a molecule such as caffeine. Now that's a small molecule and that's only going to pick up one charge. The bigger we go, the more charge we can accommodate. Okay? So I might be talking about a protein of 100,000 atomic mass units in size, and I might see, for instance, the plus 50 to plus 100 charge states of that molecule, just because of its sheer size and its ability to pick up that extra charge.